Hello there, and welcome to Sleepy Science Vault. What if the colors you see every day aren't really out there in the world at all? It sounds like a strange question. You look at a red apple, a blue sky, a green leaf, and the colors seem so obvious, so immediate, so completely real. But the deeper you look into what color actually is, the more remarkable the answer becomes. Color is not a property of objects. It isn't sitting on the surface of a flower waiting to be noticed. What objects do is absorb and reflect light, and light itself travels in waves, each with its own specific wavelength. What we call red corresponds to longer wavelengths of light, while violet corresponds to shorter ones. But here is the thing that changes everything: the wavelength itself contains no color. It is just energy, moving through space. A number. A frequency. The color only appears when that energy meets a living eye and a working brain. Which means color, in a very real sense, lives inside you. The world outside doesn't experience red or green or gold. Those are experiences, biological events, that happen within the nervous system when light strikes the eye. Two people standing in front of the same painting, bathed in the same light, may be having slightly different color experiences and have no way of ever knowing it. The colors feel absolute, but they are deeply personal. And even the range of color we can see at all is surprisingly narrow. The electromagnetic spectrum stretches across an enormous range, from radio waves long enough to span entire buildings, down to gamma rays smaller than an atom. Visible light, the thin sliver of that spectrum that human eyes can detect, occupies only a tiny fraction of the whole. Beyond violet lies ultraviolet light, which bees can see but we cannot. Beyond red lies infrared, which some snakes can sense as heat. There is an entire world of light that surrounds us constantly, moving through the air, bouncing off surfaces, and we are completely blind to it. Not because it isn't there, but because our eyes simply weren't built for it. What we call the visible world is really just the world our particular biology allows us to witness. For a long time, even the greatest scientific minds misunderstood how color actually worked. For centuries, the prevailing belief was that white light was pure and neutral, and that colors were produced when white light was somehow altered, stained or corrupted as it passed through a prism or a piece of glass. It seemed intuitive. White felt like the absence of interference, and color felt like something added. Isaac Newton changed all of that. In the 1660s, Newton took a glass prism into a darkened room, allowed a thin beam of sunlight to pass through it, and watched something extraordinary happen. The beam spread into a full band of color, red bleeding into orange, orange into yellow, yellow into green, into blue, into violet. He had seen this before, as anyone who looked through glass had. But Newton didn't stop there. He took a second prism and reversed the process, recombining the spread of color back into white light. What this proved was remarkable: white light was not simple at all. It was a mixture, a hidden complexity. All those colors were already present inside it, bundled together, only waiting for something to pull them apart. It was one of the quiet revolutions in how humans understood the world. Now, not everything we call a color fits neatly into that band of wavelengths. black and white occupy a different category entirely. The visible spectrum, that long ribbon of color from red to violet, contains no black and no white. Black is not a color of light at all. It is the absence of light, what the eye perceives when very little or no light reaches it. White, on the other hand, is the perception of many wavelengths combined, arriving together in roughly equal measure. They describe conditions of light, rather than specific wavelengths within it. And then there are colors the spectrum simply never shows you at all. Magenta, for instance, has no wavelength of its own. It doesn't exist as a single frequency anywhere in the rainbow. It is invented by the brain to bridge a gap, to reconcile the simultaneous stimulation of red and violet receptors when no single wavelength can account for both at once. The brain, rather than leaving the question unanswered, creates something new. So when you look at the world around you and see it glowing with color, every shade and hue and subtle variation, you are witnessing something that is partly physics and partly you. The light is real. The wavelengths are real. But the color, that lived and luminous experience, is something your mind is building, moment to moment, from raw information that contains no color of its own. The apple is not red. The sky is not blue. And the world, in some profound sense, would be entirely colorless, without someone there to see it. There's a color you see almost every day that does not exist anywhere in the physical world. Not in sunlight. Not in a rainbow. Not in any wavelength of the electromagnetic spectrum. And yet it appears perfectly real to you, vivid and distinct, completely unmistakable. That color is pink. Or more precisely, what scientists sometimes call magenta, that warm, slightly purple-tinged rose that sits between red and violet in your perception. If you look for it in a prism, in a rainbow, in any spread of pure spectral light, you will not find it. It simply isn't there. And the reason for that tells us something extraordinary about how the mind works. Color, as we explored before, is not a property of light itself. It is something the brain constructs from information. And sometimes, the brain is asked to make sense of a situation that has no clean physical answer, and rather than leaving a blank, it invents something. When your eyes receive strong stimulation from both red receptors and violet receptors simultaneously, with little green stimulation between them, the brain faces a kind of puzzle. These two signals are at opposite ends of the visible spectrum. They don't meet in the middle the way that yellow sits between red and green. There is no single wavelength that explains the combination. So the brain bridges the gap. It creates magenta, a color that exists only in perception, stitched together from two separate signals, a solution the visual system generates entirely on its own. In this sense, pink is not a lie. It is something more interesting than that. It is evidence of the brain's constant, invisible creativity. And pink is not alone. Many of the subtler colors we experience, the muted tones, the complex hues, the colors that don't have easy names, are combinations the visual system assembles from simpler signals. The brain is not a passive recorder of the world. It is an active interpreter, always working to turn raw data into something coherent and meaningful and complete. But to understand how any of this happens at all, We have to go deeper, past perception, past psychology, all the way down to physics. Every color you have ever seen began with the same event, light colliding with an electron. Atoms are surrounded by electrons, arranged in specific energy levels. When a photon of light strikes an atom, it can be absorbed by an electron, which then jumps to a higher energy level. Different electrons absorb different wavelengths, different amounts of energy, depending on their structure. The wavelengths that are not absorbed get reflected back into the environment. And those reflected wavelengths are what eventually reach your eyes. This is why a leaf is green. The chlorophyll molecules inside it absorb red and blue light quite efficiently, using that energy to drive photosynthesis. The green wavelengths, less useful for that process, are reflected away. Your eye catches them. Your brain reads them. And what you experience is greenness, vivid, unmistakable, real in every felt sense, even though what actually happened was a quantum interaction at the scale of individual electrons, invisible beyond all imagining. Color is physics made personal. Now, all of this extraordinary chemistry and light would be meaningless without the right biological machinery to receive it. And the human eye is, in its way, a remarkable piece of engineering shaped by hundreds of millions of years of evolution. The part of the eye responsible for color vision is found in the retina, the thin, light-sensitive layer at the back of the eye. Scattered across the retina are two types of photoreceptor cells, rods and cones. Rods are extraordinarily sensitive to light and handle vision in dim conditions, but they don't distinguish between colors. Cones are the color detectors. They require more light to function, which is why colors fade and flatten in the dark. But in good light, they bring the world to life. The human eye contains 3 distinct types of cone cells. Each type is sensitive to a different range of wavelengths. One type responds most strongly to long wavelengths, what we experience as red. Another peaks in the medium range, greens and yellows. The third is tuned to shorter wavelengths, blues and violets. These 3 types of cone don't work independently. They work together, each sending its own signal to the brain, and the brain compares those signals constantly, building color from the relationships between them. Because our eyes use 3 types of receptor to decode color, we are what scientists call trichromatic. 3 channels of information, flowing in parallel, producing the full richness of human color vision. It is an elegant system, simple in structure, vast in output. And the output is genuinely vast. Most people with typical color vision can distinguish somewhere in the region of 1 million distinct colors. Think about what that actually means. 1 million variations in hue and saturation and brightness, each one different enough to be recognized as separate from the others. The world you move through is painted in extraordinary detail, and your visual system is sensitive enough to notice nuances of color that most people would struggle to put into words. That sensitivity begins the moment light touches the cone cells in your retina. When a photon reaches a cone cell, it triggers a cascade, a chain of chemical reactions that converts the light signal into an electrical one. The cone doesn't send a message that says red or green. It sends a signal indicating how strongly it was stimulated. the three cones report their individual levels of activation, and the brain takes those three numbers and works out what color the light represents. It is a bit like mixing primary colors, only in reverse. Instead of combining pigments to create a color, the brain is decoding a color by comparing three separate measurements. It happens in milliseconds, continuously, without any effort or awareness on your part. Now here is something subtle but worth sitting with. Brightness and color, though they feel seamlessly combined in your experience of the world, are actually processed in partly separate ways inside the visual system. Your brain has dedicated pathways for detecting how light or dark something is, and other pathways more specifically tuned to its color. These two streams of information run in parallel, sometimes influencing each other, sometimes operating quite independently. This is why optical illusions involving color and brightness are so reliably disorienting. When the two systems give conflicting information, when your brightness detection pathway reads a shadow differently from how your color system reads it, the result can be genuinely confusing, even when you know intellectually what's happening. The famous illusion of two squares on a checkerboard that appear to be different shades but are physically identical works precisely because the brightness system is being fooled by context. The color information is accurate. The brightness interpretation is not. The brain is doing its best with incomplete information. It always is. This separation also helps explain something simple but often overlooked. Color adds a dimension to vision that brightness alone cannot provide. In a black and white world, you can tell that two objects are different, lighter, darker, more contrasted, less so, but you cannot tell why they differ. Color gives you more information. It tells you that this berry is ripe and that one is not, that this flower is different from the leaf beside it even when they are the same brightness, that the sky at dusk is doing something separate from the sky at noon. For a creature navigating a complex natural environment, finding food, recognizing kin, avoiding danger, that extra dimension of information is enormously valuable. Color vision, in this sense, is not a luxury. It is a tool. One that evolution refined carefully, across deep stretches of time, long before any human ever thought to wonder what a color actually was. And yet here we are, wondering. Looking at the world through these ancient, intricate eyes, watching it glow with a million shades of meaning, and asking, quietly, where all of that color really comes from. The answer, as it turns out, is somewhere between the light and the mind. Somewhere in the space between physics and experience, where the world ends and you begin. When the light fades and evening settles in, something quiet happens to the way you see the world. Colors begin to lose their richness. Reds go flat and dark. The vivid greens of the afternoon soften into gray. Everything takes on a cooler, less certain quality. Shapes remain, but the warmth drains out of them. This isn't imagination. It is biology. As light levels drop, your cone cells gradually hand over responsibility to a different set of photoreceptors entirely, the rods. Rods are built for sensitivity rather than color. They can detect extraordinarily small amounts of light, far less than the cones require, and they are what allow you to navigate a dark room, to see a path under starlight, to notice movement at the edge of vision on a moonlit night. But they deal almost entirely in brightness. In low light, the world is rendered in a kind of biological monochrome, shaded and atmospheric but stripped of its color detail. This shift happens gradually, seamlessly, and most people never consciously notice it. The visual system manages the transition quietly, the way the body manages most of its most essential functions, without asking for attention, without interrupting the experience of simply being alive and looking at the world. The fact that humans have this two-system approach, rods for low light, cones for color, is not accidental. It is the product of an enormously long evolutionary history, shaped by the specific conditions in which our ancestors lived and survived. For millions of years, the primary source of light available to living things was the sun. Natural daylight, with its particular blend of wavelengths, and the soft scattered light of dusk and dawn. The human visual system was calibrated against that light, tuned to it over vast stretches of time, long before fire, long before artificial illumination of any kind. In many ways, our eyes still expect the sun. The quality and color temperature of natural daylight is built into our perception at a very deep level, shaping not just what we see but how we feel about what we see. Which may be part of why time spent outdoors under open skies so often feels, in some hard-to-articulate way, clarifying. The light is the kind of light we were made for. Now, within that long evolutionary story, humans occupy a somewhat unusual position among mammals. Most mammals, dogs, cats, horses, deer, the great majority of the group, have only two types of cone cells rather than three. Their color vision is therefore less detailed than ours, roughly comparable to what a person with red-green color blindness experiences. They can still distinguish colors to some degree. But the rich, full-spectrum color world that humans inhabit is not available to them in the same way. This wasn't always considered surprising. For a long time, it seemed natural to assume that more complex animals would have more capable sensory systems across the board. But the reality is more interesting than that. Mammals as a group largely lost the richer color vision that earlier vertebrates possessed. The leading theory is that the common ancestor of most mammals lived through a long period as small, nocturnal creatures, animals active in darkness, for whom sensitive low-light vision was far more important than color discrimination. Under those conditions, maintaining the full machinery of rich color vision was an expensive luxury with limited practical benefit. Primates, the group that includes humans, apes, and monkeys, are among the exceptions. The trichromatic color vision that humans possess re-evolved in the primate lineage, likely as an adaptation to life in trees and forests where distinguishing ripe fruit from unripe fruit, or reading the subtle color signals in the faces of other individuals, provided a real survival advantage. The three-cone system was rebuilt, essentially, for a different way of living. But here's the remarkable thing. Primates are far from the pinnacle of color vision in the animal kingdom. Not even close. Many birds, for instance, have 4 types of cone cells rather than 3. They are tetrachromatic, operating with a 4th channel of color information that extends their perception into the ultraviolet range. What a bird experiences when it looks at another bird, at a flower, at the sky, involves color information that is entirely invisible to human eyes. The plumage patterns of many bird species, when photographed under ultraviolet light, reveal elaborate markings and signals that appear plain or uniform under the wavelengths humans can see. Birds are communicating in a visual language we are simply not equipped to read. Reptiles and fish show similarly rich color systems. Many fish have 4 or even more photoreceptor types. Some are sensitive to wavelengths well outside the human visible range in both directions. The underwater world, already so different in texture and light from the world above the surface, is rendered in colors for which we have no name and no concept, frequencies of light that have never once produced a color experience in a human mind. Color vision, it turns out, has evolved independently multiple times across the history of life. It is not a single invention passed down through all-seeing creatures. It is a solution that different lineages found separately, each time shaped by the particular challenges and environments those creatures faced. Eyes themselves evolved independently somewhere between 40 and 70 times in different animal groups, and color vision followed along various separate paths within that broader story. What this means is that vision, seeing, the act of turning light into experience, is not one thing. It is many things, arrived at by many routes, producing many different worlds of perception that overlap only partially, and sometimes not at all. Consider the butterfly. Many butterfly species possess 5 or more photoreceptor types, some extending well into the ultraviolet. The flowers they visit have co-evolved with them, developing ultraviolet patterns, guides and signals and markings that are invisible to us but strikingly clear to a butterfly's eyes. When you look at a meadow of flowers on a summer afternoon and find it beautiful in its color, you are seeing perhaps a fraction of what is actually displayed there. The meadow is dressed in patterns for an audience that isn't human, communicating in a wavelength you cannot see. And then there is the mantis shrimp. The mantis shrimp is a small, extraordinary marine creature that has become something of a legend in discussions of color vision, and for good reason. It possesses up to 16 different types of photoreceptor. 16. By any simple comparison, that sounds like a color perception of unimaginable richness and complexity. And in some ways it is. But the mantis shrimp's visual system appears to work quite differently from what we might expect. Rather than combining signals to discriminate between closely similar colors, the way human trichromacy allows us to distinguish that million-shade range, the mantis shrimp seems to use its many receptors more like a rapid scanner, quickly categorizing colors into broad groups rather than making fine distinctions between them. The result is a visual world that is alien in its structure, not just its range. Not richer in the way a painting with more paint is richer. Different in a more fundamental way, organized by different rules, built for different purposes, filtered through a completely different kind of mind. It is a reminder that there is no single correct way to see. The world does not have one true appearance that some creatures perceive clearly and others miss. Every eye is a particular conversation between physics and biology, asking specific questions about the environment and receiving specific answers. Each one illuminates a different version of the same world. Now, back among humans, there is a quiet and fascinating exception to the standard 3-cone system, one that went unnoticed for a long time, and may be more common than most people realize. Some individuals, most of them women, appear to carry a fourth type of cone cell. They are called tetrachromats, and their condition arises from a specific genetic variation on the X chromosome. Because women have two X chromosomes, they can, in certain circumstances, carry two slightly different versions of one of the cone pigment genes, one on each chromosome, giving them 4 distinct cone types rather than the standard 3. Men, with only one X chromosome, are much less likely to develop this configuration. Tetrachromacy has been studied carefully since it was first hypothesized in the 1940s, and while the genetics are well established, the perceptual consequences are still being explored. The presence of 4 cone types doesn't automatically produce richer color perception. The brain also has to develop the neural wiring to make use of that extra channel of information, and that development may depend on experience, environment, and individual variation. But at least some tetrachromats do appear to perceive color distinctions that are invisible to the rest of the population. In tests using carefully constructed color samples, colors that appear identical to trichromatic observers, certain tetrachromats can reliably tell them apart, detecting a difference where others see none. The world, for these individuals, contains more color information than the same world contains for the people standing beside them. 2 people looking at the same painting, in the same light, having genuinely different experiences, not because one is mistaken, but because they are each looking through a different kind of eye. It's a thought worth sitting with quietly for a moment. The color experience you have, the particular richness and range of the world as you see it, is not universal. It is specific to you, to your biology, to the particular set of receptors and neural pathways you were born with and have developed across a lifetime. Other humans may be seeing colors you cannot distinguish. Other species are perceiving wavelengths you will never experience. The world is filled with color that no human eye has ever registered, in frequencies that have never once, in all of human history, produced a conscious experience of color in a human mind. And yet the portion of that world that does reach us, that narrow, personal, biologically specific slice of all possible light, is still enough to fill a lifetime of looking. Enough to move us in front of a sunset. Enough to make the colors of autumn feel like something close to a gift. The universe is richer than any one pair of eyes can hold. And somehow, even knowing that, the view from where you are is still quietly, undeniably, beautiful. Human color vision is not a single, uniform experience shared equally by everyone. Even among people with otherwise typical sight, the quality and precision of color perception varies in ways that are subtle but real. Some of this variation is random, small natural differences in the density of cone cells, in the clarity of the lens, in the particular tuning of individual photoreceptors. But some of it follows a clear pattern, and that pattern runs along the X chromosome. The genes responsible for 2 of the 3 human cone pigments, the ones tuned to red and green wavelengths, are both located on the X chromosome. Because women carry 2 X chromosomes and men carry only 1, the consequences of variation in those genes fall differently across the sexes. A woman who carries a variant version of one of those pigment genes on one chromosome often has a typical version on the other, and the typical version is usually sufficient to produce normal color vision. The variant is masked. A man with a variant version on his single X chromosome has no backup. Whatever that gene produces is what he works with. This asymmetry has some interesting consequences. One of them is the possibility of tetrachromacy, which we explored in the last section, the four-cone condition that arises in some women who carry two slightly different versions of the same pigment gene. But another consequence runs in the opposite direction, toward reduced color discrimination rather than enhanced. And it is far more common than most people realize. Among people with what is called red-green color vision deficiency, some do not experience the typical 3 distinct peaks of cone sensitivity. Instead, 2 of their 3 cone types have overlapping or shifted sensitivities. The red and green receptors respond to more similar ranges of wavelengths than usual, making it harder to tell them apart. The world still has color. Most colors are still distinguishable. But in the zone where red and green meet and blend, the distinctions become difficult or impossible to make. Red-green color blindness, using the common name, though the technical picture is more nuanced, is the most prevalent form of color vision deficiency in humans. Roughly 8% of men and around 0.5% of women experience some version of it. In a room of 100 people, several will be seeing color in a measurably different way from the majority around them, and in many everyday situations, nobody will know, because the difference only becomes apparent in specific circumstances. It is largely inherited, passed down through families in the pattern that the X chromosome location of those genes would predict. A father with red-green deficiency cannot pass it directly to his sons. They receive their X chromosome from their mother, but his daughters will all carry at least one copy of the variant gene, and may pass it on in turn. The pattern threads quietly through generations, appearing and disappearing, rarely causing significant difficulty in daily life but coloring, quite literally, the experience of millions of people worldwide. Beyond inherited variation, color vision can also be changed by life itself. By illness, by injury, by the specific damage that certain events leave behind in the brain. There is a condition called achromatopsia, a state in which a person loses color vision entirely, seeing the world in shades of gray without any color information at all. In its inherited form, it is present from birth, caused by the absence or dysfunction of cone cells. But it can also be acquired. It can arrive suddenly, as the result of damage to specific regions of the brain, leaving a person who once saw full color unable to perceive it at all, not because their eyes have changed, but because the part of the brain that processed color has been disrupted. This is one of the clearest signs that color is not made in the eye. The eye collects light. The retina converts it into electrical signals. But the experience of color, that vivid, felt quality of redness or blueness or the particular warmth of amber, is assembled somewhere else. It is assembled in the brain. And the journey from eye to experience is longer and more complex than most people ever imagine. When light falls on the cone cells in your retina, the photoreceptors respond and generate electrical signals. Those signals travel along the optic nerve, a dense bundle of nerve fibers carrying information away from the eye toward the rest of the brain. The two optic nerves, one from each eye, meet at a point beneath the brain called the optic chiasm, where information from the left and right visual fields is sorted and redirected. From there, the signals travel deeper into the brain, reaching a structure called the lateral geniculate nucleus, or relay station tucked within the thalamus, the brain's central hub for sensory information. At this relay station, the signals are organized and processed before being sent onward to the visual cortex, a large region at the back of the brain dedicated to making sense of what the eyes have seen. But even within the visual cortex, the processing doesn't happen in one place all at once. Different aspects of the visual scene are handled by different specialized regions, each attending to its own part of the problem. One region focuses on edges and boundaries. Another handles motion, the direction and speed of things moving through the visual field. Another deals with the recognition of shapes and objects, building up from simple features to complex wholes. And certain areas are specifically dedicated, or at least specifically important, to the processing of color. This specialization was discovered partly through cases of brain damage. A person who suffers damage to a region called V4, located in the ventral stream of the visual cortex, may develop what is known as cerebral achromatopsia, color blindness caused not by any problem with the eyes themselves, but by the loss of the brain tissue that gives color signals their meaning. The eyes still work. The retina still responds to different wavelengths differently. The signals still travel along the optic nerve. But at the point where those signals should be transformed into the experience of color, the machinery is broken. The signals arrive and find nowhere to go. Some people with cerebral achromatopsia describe the experience vividly. The world looks, they say, like a very poor quality black and white film, flat and washed and strange. Not dark, not blurred, just drained. They know intellectually that the sky should be blue and that grass should be green, but they cannot experience those colors anymore. The knowledge of what color was remains. The experience of it is simply gone. It is one of those neurological realities that quietly reframes everything. Color is not a fact about the world that the eye reads off and reports. It is a construction. A product of neural processing. Something the brain makes from raw measurements that don't, in themselves, contain it. The signals traveling up the optic nerve don't carry labels that say red or teal or golden. They carry numbers, levels of activation in 3 different cone types, and the brain works out the rest. It compares signals across the retina, weighing local contrasts against broader context. It compensates for changes in lighting, making adjustments that allow a red apple to look red whether it is lit by noon sunlight or the warm amber of a late afternoon. It factors in memory, expectation, and the colors of surrounding objects, all without any conscious instruction from you. This compensatory process is called color constancy, and it is one of the visual system's most elegant achievements. The wavelengths of light reflected from a surface change dramatically depending on the light source illuminating it. A white piece of paper reflects very different wavelengths indoors under electric light than it does outside under open sky. But to you, the paper looks white in both situations. The brain is quietly correcting for the change in illumination, holding the perceived color of objects stable even as the physics of the light shifts constantly around them. Without color constancy, the world would be a bewildering and unreliable place, every change in cloud cover or time of day sending colors lurching into new and unfamiliar territory. Instead, most objects look more or less the same color in most conditions, which is enormously useful for a creature that needs to recognize things reliably across a wide variety of lighting situations. But color constancy is also the source of one of the most famous perceptual phenomena in recent memory. In 2015, a photograph of a dress circulated on the internet and divided the population in a way that seemed almost impossible. Some people looked at it and saw a blue and black dress. Others looked at exactly the same image and saw white and gold. The disagreement was total and genuine, not a matter of different monitors or different viewing conditions, but of different brains making different assumptions about the light source in the scene. The visual systems of the white and gold group were interpreting the image as if it were lit by a warm, yellowish light, and correcting accordingly, stripping out the assumed warm cast and reading the dress as lighter and more golden than the raw pixel values suggested. The blue and black group were making a different assumption about the lighting and correcting in the opposite direction. Both groups were doing exactly what the visual system is supposed to do. They were just doing it differently. The dress itself had one set of physical properties. The color experiences it produced were genuinely, measurably different. What this illustrates, what it makes viscerally real in a way that abstract explanations sometimes cannot, is that your visual system is never simply recording reality. It is always interpreting it. Always making assumptions. Always building the most plausible version of the world it can from the signals available, filling in gaps, resolving ambiguities, constructing a coherent picture from incomplete information. Color is part of that construction. A crucial part, rich with information and meaning. But a construction nonetheless. The visual journey from photon to experience involves the retina, the thalamus, and multiple regions of the cortex, each contributing something essential. Damage any link in that chain and the experience changes, colors dim, shift, simplify, or disappear entirely. The eye is the beginning. But experience is made further in the soft and extraordinary tissue of a brain doing what brains do best, turning the raw material of the world into something that can be felt, remembered, and understood. And somewhere in that long, layered process, somewhere between the quiet arrival of light on a receptor cell and the moment you simply, effortlessly see, color appears. Not in the light. Not in the object. Not even exactly in the eye. But in the place where all of it finally comes together, where physics meets biology meets mind, in the only place experience has ever happened, or ever could. In you. By the time a color reaches your awareness, it has passed through a remarkable number of stages. It began as light, a particular mixture of wavelengths reflected from a surface and traveling through air toward your eye. It was absorbed by cone cells, converted into electrochemical signals, sorted and relayed through the lateral geniculate nucleus, and sent onward to the visual cortex. And even there, in the cortex itself, the processing is not finished. It continues across multiple specialized regions, each one attending to a different piece of the problem, each one contributing something that the others cannot provide alone. The brain's approach to color is not simple or linear. It is distributed, spread across several overlapping systems that work in parallel, checking each other's work, combining their outputs into something unified and coherent. Understanding even a little of how this works reveals just how much invisible effort underlies the effortless act of simply opening your eyes and seeing the world in full color. One of the most important early steps in this process happens at the level of what are called opponent color channels. The raw signals from the three cone types, roughly red, green, and blue, don't travel directly to higher brain areas in their original form. Instead, they are recombined almost immediately into contrast signals, red versus green, blue versus yellow, and light versus dark. The brain doesn't record how much red and how much green are present independently. It records the difference between them. This opponent coding is extraordinarily efficient. It captures the most useful color information in the fewest possible signals, compressing the three cone measurements into something the brain can work with more easily. And it reflects something deep about how color functions in perception. What matters, most of the time, is not the absolute wavelength of light hitting a surface but how that surface differs from its surroundings. A red object in a red-lit room still looks red partly because its redness differs from the object surrounded in specific ways. The opponent system is built to detect those differences, to pull color contrast out of the scene even when the overall lighting shifts. Further along in this process, the visual system relies on a class of neurons called double opponent cells. These cells, found in the visual cortex, are sensitive not just to color contrast at a point in the image but to color contrast across a region. They respond when one color is present in the center of their receptive field and a different, opponent color is present in the surrounding area. They are, in a sense, looking for color edges, places where one color meets another. Double opponent cells are part of what gives color perception its stability. They help the visual system resist being fooled by gradual changes in illumination, because a gradual shift in the color of the light affects all parts of the scene somewhat equally. It doesn't create strong local color contrasts. It is the sharp color differences between objects, the kind that double opponent cells are sensitive to, that tend to remain stable and reliable even as overall lighting conditions change. These cells are part of the mechanism behind color constancy, quietly working to keep the world looking like itself across the shifting light of a full day. But color in the brain is not only about stability and accuracy. It is also about function, about what color is for. One of the most important things color does for the brain is help identify objects. The visual world is enormously complex. Every scene contains dozens or hundreds of separate objects, overlapping and occluding each other, varying in size and distance and orientation. Parsing all of that, deciding where one object ends and another begins, is a surprisingly difficult computational problem. Color helps solve it. When two adjacent surfaces have similar brightness but different colors, the boundary between them becomes visible in a way that brightness alone would not reveal. A brown branch against a brown tree trunk might be almost invisible in a black and white image, merged into the same tone. Add color and the distinction becomes immediately clear. The visual system uses color as one of its primary tools for what is called scene segmentation, the process of dividing the visual world into discrete, meaningful regions that can be recognized, tracked, and reasoned about. Consider how much harder it would be to navigate a farmer's market, or a forest, or even an ordinary kitchen, if everything were rendered in shades of gray. The individual objects would still be there. Their shapes would still be present. But the ease and speed with which you pick one thing out from another, the way your attention is guided almost automatically by color contrast, would be gone. Shopping would require more effort. Finding a ripe piece of fruit among unripe ones would take deliberate examination instead of an instant glance. The world would still be navigable, but it would demand more from you. Color reduces that cognitive load. It makes the visual scene easier to parse, faster to understand, and more reliably segmented into the meaningful units that allow you to interact with it effectively. In this sense, color is not decorative. It is structural, woven into the way the brain organizes its model of the world. And yet, for all its structural importance, for all the precision and sophistication of the neural machinery that produces it, color perception is not entirely the same from one person to the next. This is a thought that tends to feel faintly unsettling when you sit with it for the first time. The colors you see, the particular quality of the red in a stop sign, the exact shade of blue in a summer sky, feel absolutely certain to you. They feel like facts about the world, not interpretations. They feel as objective as the temperature of water or the weight of a stone. And yet the evidence suggests that what feels certain and objective in color perception is at least partly personal. The differences begin in the hardware. Cone cells vary slightly between individuals, in their density across the retina, in the precise wavelengths they respond to most strongly, in the relative number of each type. One person might have a slightly higher ratio of red-sensitive cones to green-sensitive cones than another. The proportions vary more than most people realize, and different cone ratios produce slightly different color signals from the same physical light. Small differences in the pigment molecules inside the cones can shift their peak sensitivities by a few nanometers, subtly changing the tuning of the entire system. Then there is the lens of the eye, which yellows slightly with age, filtering out some of the shorter wavelengths and changing the color balance of the light that reaches the retina. A young person and an older person looking at the same blue sky are receiving light that has been filtered to somewhat different degrees before it ever reaches a photoreceptor. Their brains will compensate. To some extent, the color constancy mechanisms work on this kind of filtering too, but the compensation is not perfect, and the underlying shift is real. Cultural and linguistic factors add another layer. There is a considerable body of research suggesting that the language you speak, and specifically the color terms your language provides, can influence how easily and quickly you distinguish between colors. Languages differ in how they carve up the color spectrum into named categories. Some languages have a single word for what English speakers call blue and green. Others make distinctions that English does not, dividing what English speakers would call a single color into two distinct named categories. And when colors fall on one side or the other of a named boundary in your language, you tend to be faster at distinguishing them than when they fall within the same named category. Color categories, it seems, are partly perceptual and partly linguistic, shaped by both the biology of the eye and the particular vocabulary you grew up with. The boundary between blue and green in your experience of the world is not drawn by physics alone. It is drawn partly by the language you use to talk about what you see. And yet, and this is what makes the whole picture so genuinely fascinating, despite all of this variation, color perception across humans is also surprisingly consistent. The differences are real, but they tend to be subtle. Most people with typical color vision agree closely on which things are red, which are green, which are blue. They agree on which colors look warm and which look cool, which look saturated and which look muted. They produce remarkably similar color naming patterns when tested across cultures, even cultures with quite different color vocabularies. The hardware varies, the linguistic labels vary, the fine discriminations at the boundaries vary, but the broad structure of human color experience is shared, stable, and recognizable across an enormous range of individual difference. Why should this be? Partly because the underlying biology is shared. The same three-cone pigments, tuned to similar wavelengths, are present in virtually all humans with typical color vision. The same opponent processing, the same color constancy mechanisms, the same cortical architecture, these are the common ground on which individual variation plays out. The variations are like small differences in the tuning of instruments in the same orchestra, real, measurable, sometimes audible in close listening, but not enough to make the music unrecognizable. There is also likely a role for shared experience. Humans have grown up, for all of recorded history and far longer, in the same world, under the same sun, surrounded by the same range of natural materials, seeing the sky and the sea and the earth and fire and living things. The colors of the natural world are the reference points against which all color perception is calibrated. We may not see them in identical ways, but we have been calibrated by the same original sources, and that shared calibration goes deep. All of which leaves us with a portrait of color perception that is neither purely objective nor purely subjective, but something more interesting than either. The physical world is real. The wavelengths are real. The cone cells are real and the neural processing is real. But the experience, the felt quality of color, the way red feels different from blue, the particular emotional weight that certain colors carry, is assembled by a brain that brings its own history, its own biology, its own linguistic and cultural context to the task. Two people can stand in the same light, looking at the same thing, and have color experiences that are genuinely, measurably similar, and also, in ways that may be impossible to fully compare, genuinely their own. We share a world. But each of us lives inside a slightly different version of it, painted in colors that are partly public and partly private, partly inherited and partly constructed, partly as old as the first light that ever fell on a living eye, and partly as new as this moment, this particular light, this particular pair of eyes, open and looking. There is something quietly astonishing hidden inside the consistency of human color perception. We have established that people differ, in their cone counts, in the precise tuning of their photoreceptors, in the linguistic categories their language provides, in the filtering effects of their individual lenses. Some people have dramatically more cone cells packed into their central retina than others. Research has found that the number of cones in the fovea, the small, densely packed central region of the retina responsible for sharp, detailed vision, can vary by a factor of 3 or more between individuals. The ratio of different cone types varies too, sometimes substantially, in ways that should, in theory, produce noticeably different color signals from the same physical light. And yet when you ask people to name colors, to sort them, to match them, to draw the boundaries between red and orange or blue and purple, the results are remarkably coherent. Not identical, there are genuine individual differences at the margins, at the boundary regions between categories, in the subtle discriminations that require careful testing to reveal. But the broad architecture of human color experience is shared in a way that the underlying variation in the hardware does not obviously predict. Something is compensating. Something is smoothing out the differences, aligning the outputs of different biological systems into something that functions, for most practical purposes, as if it were universal. That something is the brain. The visual system doesn't just process color passively, feeding raw signals upward and letting the cortex deal with whatever arrives. It actively calibrates itself, continuously, automatically, and largely without any awareness on your part. The brain monitors the overall color distribution of the visual scene, tracks the statistics of what it is seeing, and adjusts its processing to maintain consistent color output even when the input signals shift. This calibration happens on multiple timescales. On the very short scale of milliseconds, neurons adapt to strong stimulation by reducing their sensitivity, the mechanism behind afterimages, which we will come to shortly. On the scale of seconds and minutes, the visual system adjusts to the prevailing color temperature of the light source, recalibrating its sense of what counts as neutral white and working outward from there. And there is evidence that on longer timescales, days, weeks, the system can recalibrate more deeply, adjusting to persistent changes in the color statistics of the environment. This longer-scale recalibration is beautifully illustrated by what happens when people wear tinted lenses for extended periods. In the short term, colored lenses shift the appearance of everything. The world looks warmer, cooler, more reddish, more greenish, depending on the tint. The effect is immediate and obvious. But over days and weeks of continuous wear, the visual system gradually adapts. The tinted cast fades. The world begins to look more normal, more natural, more like the unfiltered world the brain remembers. The lens hasn't changed. The physics of the light reaching the eye hasn't changed. The brain has simply recalibrated its baseline, accepted the new color statistics of its environment, and adjusted its output accordingly. When the lenses are removed after this adaptation period, the reverse effect appears. The world briefly looks tinted in the opposite direction, until the system recalibrates back. The brain is always seeking a stable, internally consistent model of color. It will correct for almost anything to maintain that stability, given enough time and consistent enough input. This is why people with dramatically different numbers of cone cells can still, for the most part, agree on what color something is. The brain compensates for the variation in hardware, normalizing the output to fit a shared framework of color experience that transcends the individual differences in the machinery producing it. It is an extraordinary piece of biological engineering, a self-tuning system that maintains coherent color perception across a population of individuals whose eyes are all, in detail, slightly different. But this same capacity for calibration and correction, this tendency of the visual system to impose its own assumptions on raw sensory data, is also the source of color's most fascinating and sometimes disorienting behaviors. Because a brain that actively interprets and adjusts is also a brain that can be fooled. And the visual system, for all its sophistication, is fooled by color with remarkable reliability. The most fundamental of these phenomena is color constancy itself, which we have touched on before, but which rewards a closer look. The light that illuminates the world changes constantly and dramatically across a single day. Dawn light is warm and golden, rich in long wavelengths. Midday sunlight is cooler and bluer. Overcast light has a different quality again. Candlelight is deeply warm, shifting the colors of everything it touches toward orange and amber. Under each of these light sources, the wavelengths reflected from a surface are genuinely different. A white wall under candlelight reflects far more long wavelength light than a white wall under an overcast sky. The physics are unambiguous. And yet the wall looks white in both situations. Not slightly different whites, white. The same white. The brain is performing a continuous correction, estimating the color of the illumination and dividing it out, extracting what it takes to be the true color of the surface from the combination of surface and light. Most of the time, this process is so smooth and automatic that we are completely unaware of it. The world just looks like itself, bathed in changing light but holding its colors steady. Color constancy is one of the visual system's great achievements. But it is not perfect, and the conditions under which it fails reveal just how much interpretive work is quietly happening beneath the surface of ordinary seeing. Context is perhaps the most powerful force shaping color perception, and its effects are far more dramatic than most people realize until they encounter a good demonstration. The same color, the same physical wavelengths, the same objective stimulus, can appear to be entirely different colors depending on what surrounds it. This is not a subtle effect at the edge of perception. It can be enormous. A mid-gray square placed on a black background appears noticeably lighter than the same gray square placed on a white background, even though both squares are physically identical. This is simultaneous brightness contrast, and color works the same way. A patch of brownish-orange on a dark background can appear as a vivid, glowing orange, a completely different perceptual quality, even though the patch itself is unchanged. The visual system is not measuring the absolute color of a surface. It is measuring it relative to its surroundings, always asking not what wavelengths are here, but how does this compare to what is around it? This comparative processing is generally extremely useful. It is part of why color constancy works, part of why you can identify objects reliably across changing lighting. But it also means that the perceived color of any surface is not determined by that surface alone. It is determined by the surface in context. Change the context, and you change the color. Nearby colors bleed into each other in perception. A gray patch surrounded by red will take on a faint greenish cast, a simultaneous color contrast effect, the opponent system generating a mild opposite response to the strong color signal in the surround. A muted, brownish color surrounded by vivid, saturated colors will appear even more muted and desaturated by comparison. A color surrounded by similar hues will look more distinctive and separate than the same color embedded in a field of contrasting ones. These are not illusions in the colloquial sense. They are not tricks or failures. They are the visual system doing exactly what it is supposed to do, working as designed, applying the principles of relative comparison and contextual interpretation that make it so effective in natural settings. The failures only become visible when those principles are tested in artificial, controlled conditions, when someone constructs a demonstration specifically designed to reveal the gap between the physical stimulus and the perceptual experience. And those demonstrations are, it must be said, genuinely startling when you first encounter them. Some of the most compelling involve what are known as simultaneous contrast illusions, where two physically identical color patches appear obviously and unmistakably different because of the colors surrounding them. The classic checkerboard shadow illusion, in which two squares of identical gray appear to be very different shades because one sits in shadow and one in light, is perhaps the most famous. What makes it so remarkable is that knowing about the illusion does nothing to dispel it. You can measure the squares. You can cover the surrounding area. You can convince yourself intellectually, beyond any doubt, that the two squares are identical. And then you uncover them, look at the whole image, and they look completely different again. This is important. It is not a matter of being naive or inattentive. The perceptual system and the cognitive system are operating separately, and the perceptual system is not taking instruction from the cognitive one. The brain that knows the squares are the same gray is not the same process as the brain that sees them as different. Both are running simultaneously, and they cannot fully override each other. It is a small but profound reminder of the architecture of mind, the way conscious knowledge and perceptual experience operate on different tracks, influencing but not fully controlling each other. You can know something and still see something different. The seeing and the knowing are not the same thing, and in color perception, that gap is sometimes wide enough to be genuinely humbling. There is also the phenomenon of chromatic adaptation, the way the visual system adjusts to a persistent color in the environment, recalibrating over time and then revealing the adjustment when conditions suddenly change. Stare at a strongly saturated color for 30 seconds, a vivid red, say, and then shift your gaze to a neutral gray surface. What you see is not gray. You see a patch of soft green, the opponent color, floating ghost-like over the surface, gradually fading as the adaptation dissipates. This is an afterimage, and it arises because the red-sensitive part of your visual system has been fatigued by the sustained strong signal. When the signal is removed, the green-sensitive pathway briefly dominates, and the brain reads the imbalance as color. Afterimages are fleeting, but they reveal something permanent about the structure of color vision, that it is always relative, always comparative, always calibrated against a moving baseline that shifts with experience and exposure. The color you see at any moment is not determined solely by the light arriving in that moment. It is shaped by the light that arrived before it, by the recent history of stimulation that has left its traces in adapted and readjusted neurons across the visual system. You carry the recent past inside your perception, quite literally. The light of a moment ago is still influencing the color of right now. There is something almost poetic in that. in the way perception is never quite a clean, fresh recording of the present moment, but always a layered thing, built from now and shaped by before, constructed from both the world outside and the history of a particular pair of eyes moving through it. Color is stable enough to be useful, consistent enough to be shared, reliable enough to anchor our experience of the world. And at the same time it is personal, contextual, dynamic, and endlessly shaped by the brain's invisible interpretive work. It is one of those aspects of experience that seems, at first glance, to be simply a fact about the world, and turns out, on closer examination, to be something far stranger and more wonderful. A fact about the world, yes, but also a conversation between the world and the mind that sees it. Always ongoing. Always adjusting. Always, in some small and unnoticed way, new. There is a peculiar fact about color that reveals, more clearly than almost anything else, just how much the experience depends on the observer rather than the object. Two light sources can be physically, measurably different, emitting entirely different mixtures of wavelengths, and yet produce an experience that is completely, indistinguishably identical. Not similar. Not close. Identical. You could not tell them apart by looking, no matter how carefully you looked, no matter how long you stared. And yet if you measured them with instruments, they would be clearly, objectively different. This phenomenon is called metamerism. It arises directly from the architecture of human color vision, from the fact that we have only 3 types of cone cells, each responding to a broad range of wavelengths rather than a single precise one. What the visual system receives is not a detailed breakdown of all the wavelengths present in the light. It receives 3 numbers, the activation levels of the 3 cone types. And crucially, many different physical combinations of wavelengths can produce the same 3 activation levels. Different causes, identical effect. Different light, identical color. This is why color matching in manufacturing and design is such a careful and sometimes frustrating science. A paint color matched under one type of lighting, the cool fluorescent light of a factory, say, may look noticeably different from its intended match under the warmer light of a home interior. The 2 surfaces are metameric under factory light. Their different pigment mixtures happen to stimulate the 3 cone types equally under that particular illumination. Change the light source, shift the wavelengths, and the metameric relationship breaks down. The same paint, the same surface, the same eye, but a different light, and the match is gone. Metamerism is not a flaw in human vision. It is a direct consequence of the compression that makes color vision possible at all. Reducing the continuous spectrum of visible light to 3 numbers is what allows a compact, efficient system of 3 cone types to produce the rich range of color experience we enjoy. The cost of that compression is the occasional collapse of different physical realities into the same perceptual experience. The benefit is everything else. Now, the 3-number system that produces metamerism also gives rise to something more surprising: the existence of color combinations the visual system is structurally prevented from producing. Not colors that are hard to imagine, or colors outside our visible range, but colors that our neural architecture makes literally impossible to experience under normal conditions. The opponent process system, The way cone signals are recombined into red versus green and blue versus yellow contrast channels means that certain combinations are inherently contradictory. The red-green channel either signals red or green. It cannot signal both simultaneously. It is a single channel with two opposing directions, and a signal cannot travel in both directions at once. The same is true of the blue-yellow channel. This means that reddish-green, not olive, not brown, not some murky blend, but a color that is simultaneously both red and green in the way that reddish-blue is purple, is not something you can see. Your visual system cannot construct it. The neural machinery simply doesn't have a mode for it. Similarly, a color that is simultaneously bluish and yellowish in a direct, unblended way is outside the space of normal human color experience. These are sometimes called forbidden colors, and their existence as a logical impossibility within normal vision has fascinated researchers. There have been experiments in which scientists have attempted to induce these colors using carefully controlled visual stimulation, stabilizing an image on the retina to prevent the normal shifts that allow opponent channels to maintain their contrast processing, and some subjects have reported experiencing colors they describe as deeply strange, colors for which they have no name and no reference, colors they say are entirely unlike anything in normal experience. Whether these reports reflect genuine perception of impossible colors or some artifact of the unusual experimental conditions is still debated. But the fact that the question can even be asked reveals something interesting about the structure of color space, that it has edges, boundaries, regions that normal perception never enters. Most of the time, of course, we operate well within those boundaries, inhabiting the full familiar range of color experience without approaching its edges. And within that range, the visual system is constantly being shaped and adjusted by forces that extend well beyond the physics of light. Afterimages are perhaps the most direct demonstration of this. We have touched on them briefly, but they are worth dwelling on a little longer, because what they reveal is not just a curiosity but a window into the continuous adaptive work happening inside the visual system at every waking moment. When you stare at a colored surface for an extended period, 15, 20, 30 seconds, the neurons responsible for processing that color gradually tire. They continue to fire, but with diminishing enthusiasm, their response reducing as the sustained stimulation depletes the chemical resources driving it. This is neural adaptation, and it happens in sensory systems throughout the body. Touch a textured surface and attend closely to the sensation. Within seconds, the feeling becomes less vivid, the texture less insistent. Sustained stimulation becomes background. The nervous system is built to prioritize change, to notice what is new and different, and to quiet its response to what remains constant. In color vision, this adaptation leaves a trace. When you shift your gaze from a strongly colored surface to a neutral one, the adapted neurons are briefly underperforming. The opponent channel that was processing, say, red has been suppressed. The green side of that channel now briefly dominates, and the brain reads the imbalance as color. You see green where there is no green, floating softly against the neutral surface, slowly fading as the neural balance restores itself. The afterimage is literally the shape of your recent visual history, projected onto the present. For a few seconds, the past is visible. Illusions of color extend well beyond afterimages, and the breadth of them reveals something important, that color perception is not simply a bottom-up process, driven purely by the signals arriving from the eye. It is also shaped, substantially and demonstrably, from the top down, by what the brain expects, by what it remembers, by the context and meaning it brings to what it sees. Expectations, it turns out, influence color perception in ways that are measurable and sometimes striking. When people are shown an object in an unusual color, a blue banana, a gray strawberry, and asked to adjust the color wheel until the object looks neutral and achromatic, they tend to adjust past the actual point of neutrality in the direction of the object's expected color. The gray strawberry, to look truly gray, has to actually be slightly shifted toward green, because the brain is adding an expected reddish tint, and compensating requires correcting against that expectation. The brain is not just seeing what is there. It is seeing what it expects to be there, and the two blend together in a way that is difficult to fully separate. Memory has a similar influence. The remembered colors of familiar objects, the particular green of grass, the orange of a ripe pumpkin, the golden brown of freshly baked bread, are typically somewhat more saturated than those objects actually appear in ordinary viewing conditions. We remember colors as richer than they are. And those memories pull perception slightly toward them, creating a world that looks, in a small but consistent way, more vivid and colorful than strict measurement would confirm. This is not a distortion to be corrected. It is part of what color experience is. Memory and expectation are woven into perception so thoroughly that trying to separate them, to identify some pure, uncontaminated color experience stripped of all prior knowledge, is probably an impossible task. Every time you look at a tomato and see red, you are bringing a lifetime of tomato seeing with you. The redness you experience is partly in the light and partly in that history. Art has understood this for centuries, even without the neuroscientific framework to explain it. Painters working in the tradition of color theory, from the Impressionists exploring how colors change in different lights to the color-field painters of the 20th century filling canvases with carefully chosen expanses of pure hue, were in their way conducting experiments in perception, using the canvas as a laboratory for exploring how colors interact, how context shapes experience, how the same pigment can look like completely different colors depending on what surrounds it. Joseph Albers, the painter and educator who devoted much of his career to the study of color interaction, spent decades producing works in which simple geometric forms, typically squares nested within squares, demonstrated how profoundly context reshapes color perception. In his series Homage to the Square, the same color appears again and again in different surroundings, looking different each time, demonstrating with quiet rigor that color as experienced is always a relational thing. It cannot be pinned down. It cannot be fully known in isolation. It only exists, fully, in relation to everything around it. Language adds yet another dimension to this complex picture. The categories that language provides, the named colors, the words that mark the boundaries between one color region and another, genuinely influence how we perceive and discriminate colors, not just how we talk about them. Colors that fall on either side of a named linguistic boundary are easier to distinguish than colors that fall within the same named category, even when the physical distance between them is equivalent. Having a name for a color makes it, in a measurable sense, easier to see as distinct. This effect is particularly striking in cultures and languages that draw color boundaries differently from English. Russian, for instance, has separate basic terms for light blue and dark blue, голубой and синий, where English uses a single word for both. Russian speakers are faster and more reliable at distinguishing light blues from dark blues than English speakers, particularly when the distinction has to be made across a linguistic boundary. The word is doing perceptual work. The category is sharpening the ability to see. And then there is the periphery, the outer edges of your visual field, where your attention rarely goes but your vision never fully stops. Most people assume that their color vision extends fairly uniformly across their entire visual field. After all, the world looks colored everywhere you look. But the distribution of cone cells across the retina is dramatically uneven. The fovea, the small central region, is densely packed with cones and produces rich, detailed color vision. Moving outward from the center, cone density drops steeply. The peripheral retina has relatively few cone cells and is dominated by rods, which handle brightness but not color detail. This means that in your peripheral vision, color perception is considerably weaker than you would likely guess. Objects in the far periphery are seen largely in terms of brightness and motion. The rod-dominated system is doing most of the work. Color is present to some degree, but it is muted, less certain, more easily confused. If someone showed you a colored object far to the side of your vision, you might detect that it was there, might even detect some sense of its brightness, before you could reliably identify its color. You generally don't notice this because your eyes are always moving. Every fraction of a second, tiny eye movements called saccades sweep the fovea across the scene, rapidly building up a detailed central vision image of everything you attend to. The brain stitches these samples together into what feels like a seamless, uniformly detailed field of vision. The peripheral weakness is hidden behind the constant motion of the eye, plastered over by a visual system that convincingly presents a unified experience of a colorful world stretching from edge to edge. What you experience as your visual field is partly real and partly constructed, a composite, an edited version, assembled from rapid sampling and intelligent filling in. The periphery looks colored because the brain expects it to be colored, because the central samples have established what colors are present in the scene, and because the visual system's model of the world fills in the expected content where detailed sampling has not occurred. Which brings us back, quietly, to the idea that is wound through all of this, that seeing is never simply recording. It is always, in some measure, a creative act. The brain is not a camera. It is an interpreter, a storyteller, a constructor of experience, one that takes incomplete, noisy, constantly shifting sensory data and turns it into the vivid, stable, richly colored world you inhabit every moment of your waking life. That world feels immediate and given, as solid as the ground beneath you. But it is, in the most literal and scientifically precise sense, something your brain is making. Moment to moment. Continuously. With extraordinary skill and almost complete invisibility. The colors you see are real. And they are yours. Both things are true, at the same time, and neither cancels the other out. Most people move through their entire lives without ever noticing that a large portion of what they see is, in terms of color detail, surprisingly sparse. The peripheral weakness we explored, the way cone cells thin out toward the edges of the retina, leaving the outer visual field largely to the rod-dominated, color-poor system, goes unnoticed not because it is subtle but because the brain is so effective at concealing it. The constant sweep of eye movements, the visual system's confident filling in of expected color, the seamless model the brain constructs of a uniformly detailed world, all of it works together to hide the gap between what the eye actually captures and what the mind experiences as seeing. It is a small reminder, before we step back from the interior world of neurons and perception, that the gap between physical reality and experienced reality is always present. Always quietly there. Even in the most ordinary moments of looking at the world around you. And the physical world, it turns out, has its own extraordinary relationship with color, one that is just as rich and strange as anything happening inside the visual system. Because color in the external world is not simply a neutral fact either. It arises from specific physical processes, each one with its own elegant mechanism, each one producing color through a different means. Take the ocean. On a clear day, the sea appears blue, a deep, shifting, luminous blue that has inspired painters and poets and travelers across the whole of human history. The obvious assumption is that the ocean is simply reflecting the blue sky above it. And reflection does play a role. But it is not the whole story, and the rest of the story is more interesting. Water itself absorbs light. Not equally across all wavelengths, it absorbs longer wavelengths, the reds and oranges and yellows, more strongly than it absorbs shorter ones. In shallow water, this effect is barely noticeable. But as depth increases, the longer wavelengths are progressively filtered out. Red light is absorbed within the first few meters. Orange follows. Yellow fades further down. By the time you reach significant depth, almost all the remaining light is in the blue and blue-green range, the wavelengths water absorbs least efficiently. This is why deep water looks blue even on a gray day when there is no vivid sky to reflect. The blueness is not borrowed from the sky. It is the ocean's own, the color that remains when everything else has been taken away. It is, in a sense, the color of water itself, the color of what survives the journey through depth. The same principle operates, on a larger scale, in the color of the sky. The atmosphere scatters shorter wavelengths of sunlight, the blues and violets, more effectively than longer ones. This is Rayleigh scattering, and it is why the sky overhead appears blue during the day. Sunlight entering the atmosphere has its blue component scattered in all directions, filling the whole sky with diffuse blue light. At sunset, when the sun is near the horizon and its light must travel through a much greater thickness of atmosphere to reach your eye, the blue wavelengths have been scattered away almost entirely, and only the longer reds and oranges and golds survive the journey. The color of the sunset is what is left after the rest has been lost. Every color in the natural world has a story like this. A physical mechanism. A reason. Rainbows are among the most visually dramatic of these, and their mechanism is one of the most beautiful examples of optics in everyday experience. When sunlight enters a spherical water droplet, it refracts, it bends, Because light changes speed when it moves from air into water, and different wavelengths bend by slightly different amounts. The light then reflects off the back inner surface of the droplet and exits through the front, refracting again as it re-enters the air. Because each wavelength bends differently, they exit the droplet at slightly different angles, red at a shallower angle, violet at a steeper one, and the white sunlight is spread into its component colors, just as Newton's prism spread it, but this time by millions of individual water droplets suspended in the air. Each droplet contributes only a single color to what you see. The red you observe in a rainbow is coming from droplets at a specific angle relative to your eye and the sun. The violet is coming from a different set of droplets at a slightly different angle. The arc you see is the locus of all the droplets at exactly the right angle to deliver their particular color to your particular eye, from the particular position where you are standing. Which leads to one of the most quietly remarkable facts about rainbows: they are not arcs. They are circles. The full geometric form of a rainbow is a complete ring of color, centered on a point directly opposite the sun from your perspective, the antisolar point. What you see as an arc is the portion of that circle that rises above the horizon. From the ground, the horizon cuts off the lower half of the circle, and you see only the arch. But from an aircraft, above the cloud layer, with water droplets both above and below, a rainbow can sometimes appear in its full circular form, a complete ring of color hanging in the air, uncut by any horizon. The rainbow you see is yours alone. Because it depends on the precise angle between you, the sun, and the droplets, someone standing a few meters away is seeing a different rainbow, different droplets at the correct angles for their position. No two observers ever share a rainbow. Each one is a private optical phenomenon, assembled for a single viewpoint, existing at the precise intersection of physics and a particular pair of eyes. There is something fitting about that. The natural world produces color through mechanisms quite different from the fluorescent and manufactured colors of modern life, and the contrast is worth noticing. Neon colors, those vivid, almost impossibly bright pinks and greens and oranges found in safety equipment, highlighters, and certain textiles, achieve their particular quality through a process called fluorescence. Fluorescent materials absorb ultraviolet light, which is invisible to the human eye, and re-emit it as visible light at a longer wavelength. Under ordinary daylight, which contains a component of ultraviolet, a fluorescent surface is effectively producing its own light in addition to reflecting the visible wavelengths around it. It is emitting more visible light than it receives in the visible range, by converting invisible ultraviolet into visible color. This is why fluorescent colors appear to glow, to have an inner luminosity that non-fluorescent colors lack. They are genuinely brighter, in the measured sense, than a purely reflective surface could be. Under ultraviolet light alone, they become dramatically vivid, which is why certain fabrics and materials seem to come alive under black light. The color is not simply reflected. It is generated. Chemical reactions add another layer to the color of the world. Many of the colors in living systems arise from molecules that change their light-absorbing properties when they undergo chemical transformations. The browning of a cut apple, the fading of a colored fabric in sunlight, the development of a photograph in a darkroom, the changing color of autumn leaves as chlorophyll breaks down and the yellow and orange pigments that were always present beneath it are finally revealed. All of these are chemical events expressing themselves as color change. The color is the visible signature of a reaction, the evidence written in wavelengths that something has changed at the molecular level. Autumn leaves deserve a moment of particular attention here, because the story they tell is somewhat counterintuitive. The yellows and oranges of autumn foliage are not colors that appear as the leaves change. They are colors that were always there, present in the leaf throughout the summer, masked by the dominant green of chlorophyll. As days shorten and temperatures drop, the tree stops producing chlorophyll and the existing molecules break down. As the green fades, the yellow and orange carotenoid pigments that were hidden beneath it all along are uncovered. Autumn's warm palette is, in a sense, a revelation, not a creation but an unveiling, the leaf showing its true colors as the season strips away what was covering them. The reds of autumn are a different story. The red and purple anthocyanin pigments are largely produced fresh as autumn progresses, synthesized in the leaf during senescence, their production triggered by bright light and cool temperatures. They may serve to protect the leaf during the period when the tree is recovering nutrients from the dying tissue. The red is new, made for the occasion, a brief, vivid final production before the leaf falls. Now, from the natural mechanisms of color, we move to the engineered ones, the ways that human technology produces color for screens and prints in the vast manufactured visual landscape of the modern world. And here, the story splits in two, because the technology of color display and the technology of color printing work on fundamentally different principles. Screens produce color with light. A digital screen, whether a phone, a television, or a computer monitor, creates color by combining tiny points of red, green, and blue light at varying intensities. These three colors are the primaries of additive color mixing, the system in which colors are produced by adding wavelengths of light together. When red and green light are combined at equal intensities, the result appears yellow to the human eye. Red and blue produce magenta. Green and blue produce cyan. And when all three are combined at equal intensities, the result is white, the full presence of all three signals producing the sensation of neutral, balanced light. When the three channels are all reduced to zero, no light is emitted and the screen appears black, not a color, but the absence of output. The black of a screen is just darkness, the screen producing nothing. This is why contrast and black level matter so much in screen quality. The black on a screen is limited by how completely the screen can suppress its own light output. The system screens use, red, green, and blue, is denoted by the abbreviation RGB, and it works because of the same trichromatic architecture that underlies human color vision. Three primaries map onto three cone types. The screen is essentially sending a precisely calibrated signal directly to the three channels of the visual system, telling each one how strongly to fire. Printing works the other way around. Ink on paper does not emit light, it subtracts it. A white page reflects all the wavelengths of the light falling on it. When ink is applied, it absorbs some of those wavelengths and reflects others, and the color you see is what remains after the ink has taken its portion away. This is called subtractive color mixing, and its primaries are the inverses of the additive ones. Instead of red, green, and blue, the primaries of subtractive mixing are cyan, magenta, and yellow, each one absorbing a third of the visible spectrum and reflecting the other 2/3. Professional printing uses these three colors, plus black, giving the system its name, CMYK, where K stands for the key black plate. Black is added as a separate ink because combining cyan, magenta, and yellow in practice produces a dark brownish gray rather than a true, rich black. The physics suggests they should combine to absorb all wavelengths equally and produce black, but real inks are imperfect, and a dedicated black ink produces deeper, sharper blacks than any combination of the color inks can manage. The gap between RGB screens and CMYK printing is one of the practical realities of working with color in design and reproduction. The range of colors a screen can display, its gamut, is quite different from the range a printer can reproduce. Some vivid screen colors, particularly intense blues and greens, fall outside what most printing processes can achieve. Designers working across both media learn to navigate this gap carefully, knowing that what looks brilliant on a screen may print as something slightly flatter and less vivid. The two systems, additive and subtractive, screen and print, are each working within the same constraints of human trichromatic vision, but exploiting those constraints in opposite ways. One speaks in light. The other speaks in shadow. Both are translations of the same underlying reality into the language that a three-channel visual system can receive and decode. And in all of this, in the ocean absorbing red, in the droplet refracting wavelengths into arcs of color, in the autumn leaf uncovering its hidden yellow, in the screen sending calibrated light signals to three types of cone cells, the same truth runs quietly through everything. Color is never just one thing. It is always physical and biological at once. Always the meeting of an external process and an internal one. Always, at its heart, a relationship between light and matter, between wavelength and receptor, between the world as it is and the mind that meets it. The ocean is not blue because it decided to be. The screen is not vivid because it contains color in the way a jar contains water. Everything that looks colored is colored because something physical is happening, and something biological is receiving it, and somewhere in the space between those two events, experience occurs. Simple to look at. Extraordinary, when you stay with it long enough to really see. Not all color in nature is made the same way. When you look at a red rose or a yellow dandelion, the color you see comes from pigment, molecules embedded in the cells of the plant that absorb certain wavelengths of light and reflect others. The color is chemical in origin, a property of the molecular structure of the pigment itself, relatively stable and consistent regardless of the angle from which you view it. Turn the rose in your hand, move around it, look at it from above or below, and the red remains essentially the same. It is produced by the same chemistry regardless of your position. But there is another way to make color entirely, one that has nothing to do with pigments, nothing to do with the chemical absorption of light. One that is, in its way, more sophisticated, more precise, and in certain respects more extraordinary than anything a pigment molecule can achieve. This is structural color, and it is responsible for some of the most visually striking phenomena in the natural world. Structural color arises not from the chemical properties of a material but from its physical architecture, the way microscopic structures arranged at the scale of light wavelengths interact with incoming light, bending it, reflecting it, and causing different wavelengths to interfere with each other in ways that produce vivid, sometimes dazzling color from what are often completely transparent or colorless materials. To understand how this works, it helps to remember that light is a wave. And waves, when they meet each other, interact. When two waves of the same wavelength arrive in phase, crest meeting crest, trough meeting trough, they reinforce each other, producing a stronger signal. When they arrive out of phase, crest meeting trough, they cancel each other out, producing nothing. This is interference, and it operates on light just as it operates on water waves or sound waves. When light strikes a surface with microscopic layered structures, thin films, repeating lattices, arrays of tiny ridges or spheres arranged at precisely controlled spacings, different wavelengths reflect off different layers and surfaces and interfere with each other in different ways. Some wavelengths are reinforced and reflect strongly. Others are cancelled and disappear. The result is color, specific colors produced by specific structural geometries, without any pigment involved at all. The wing scales of many butterflies are built this way. The Morpho butterfly, native to the rainforests of Central and South America, is perhaps the most celebrated example. Its wings are an intense, metallic blue that seems almost to generate its own light, a blue so vivid and so luminous that it is difficult to photograph accurately, because camera sensors and printing processes simply cannot capture the quality of it. And yet if you were to grind those wings to powder and examine the powder under ordinary conditions, you would find that it is not blue at all. The material itself is nearly colorless. The blue exists only when the wing is intact, when the microscopic structures are in place to do their work. The Morpho wing is covered in scales, and each scale is built from layers of ultra-thin structures, ridges and branches arranged in a precise tree-like architecture, with gaps between them measured in fractions of a micrometer. This geometry is tuned to selectively reinforce blue wavelengths and cancel others through interference. The effect is so powerful and so consistent that the blue appears across a wide range of lighting conditions, more reliably vivid than almost any pigment could achieve. What is more, structural colors produced through interference can be extraordinarily durable. Pigments fade. They are molecules, and molecules can be broken by light, by heat, by chemical exposure. The energy of ultraviolet radiation disrupts their structure, and as their molecular configuration changes, so does their light-absorbing behavior. Colors fade, yellow, shift. This is why old paintings need careful conservation, why colored plastics turn brittle and pallid in sunlight, why the brilliant dyes of ancient textiles require controlled conditions to survive. Structural color has no such vulnerability. The color is not a property of any molecule that can be degraded. It is a property of the geometry, the spacing of the structures, their arrangement and dimensions. As long as the physical architecture is intact, the color remains. This is why structural colors can persist for extraordinary lengths of time. The iridescent patches on fossilized beetle wing cases millions of years old have been found to retain their structural color. The pigments, of course, are long gone. But the geometry survived, and the color that the geometry produces is still there, still functioning, still refracting light in the same way it did before any human eye existed to see it. The natural world uses structural color widely, and the range of mechanisms it employs is remarkable. Thin film interference, the same effect that produces colors in soap bubbles and oil films on water, creates the iridescent sheen on dragonfly wings, on the feathers of hummingbirds and peacocks, on certain beetle shells, on the inside surface of abalone shells. In each case, a thin layer of material with a different refractive index from what surrounds it creates interference effects that vary with wavelength and viewing angle, producing shifting, metallic, luminous color. Diffraction gratings, surfaces with very fine, evenly spaced parallel structures, spread light into its component colors the way a prism does, producing the spectral shimmers seen in some feathers and shells. Photonic crystals, three-dimensional arrays of microscopic structures arranged with the regularity of an atomic crystal lattice, create colors by a slightly different mechanism, selectively reflecting certain wavelengths based on the periodic spacing of their elements. Some of the most vivid blues and greens in nature, in certain berries, in the feathers of some birds, in the scales of some fish, are produced by photonic crystal structures. And some structural color mechanisms achieve something that thin film interference cannot, color that remains consistent regardless of the angle from which you view it. Thin film interference is angle dependent, the color shifts as you tilt the surface, because changing the viewing angle changes the path length that light travels through the film and alters the interference condition. This is the shifting, rainbow-like quality of an oil slick, the way a soap bubble's colors swirl and change as it moves. It is beautiful, but it is also directionally variable. Certain nanostructures, arrays of tiny spheres or pores arranged with specific geometries, produce color through a mechanism that is much less dependent on viewing angle. The blue-gray of many bird feathers, including the eastern bluebird and the blue jay, arises not from thin films but from arrays of air-filled pockets or nanostructures within the feather barbs that scatter short wavelengths preferentially, a mechanism somewhat analogous to the Rayleigh scattering that makes the sky blue, operating at the scale of feather microstructure. The result is a blue that looks essentially the same from all directions, a structural blue that has the consistency of a pigment color without being chemically produced. The contrast between angle-independent and angle-dependent structural color is part of what makes the visual world of nature so varied and surprising. The consistent, matte blue of a bluebird's back and the shifting, metallic iridescence of a hummingbird's gorget are both structural colors, both produced by physical architecture rather than chemistry. But they operate through different mechanisms and produce dramatically different visual effects, one stable and clean, the other alive with movement and change, transforming with every shift in light or viewing angle. The peacock tail feather is perhaps the most familiar example of this dynamism. The extraordinary eye pattern of the peacock's display, those concentric rings of iridescent green, gold, blue, and bronze, is entirely structural in origin. The feather barbs are built with photonic crystal-like layers that produce interference colors, and as the bird moves, as the light changes, as the observer's angle shifts, the colors shift with them, rippling across the display in waves of metallic brilliance. It is one of the most visually complex natural phenomena visible to the naked eye, and it is produced by geometry alone, by the precise spacing of structures measured in nanometers, shaped by millions of years of evolutionary pressure toward ever more effective visual displays. What is extraordinary to consider is that the peahen selecting a mate on the basis of that display is, in a very real sense, evaluating the quality of a piece of nanotechnology. The regularity and precision of the photonic structures determine the intensity and purity of the colors. A male with more consistent nanostructure produces more vivid, more uniformly colored plumage. The female's preference for bright, well-defined structural color is, whether or not either bird has any awareness of this, a preference for precision engineering at the nanoscale. Evolution, operating over sufficient time, builds things that rival the most sophisticated technology humans have yet produced, and does so by selection alone, without foresight or intention, simply by preserving what works and discarding what does not. Some structural colors have properties that extend well beyond their visual impact in ordinary daylight. Certain structural color systems remain effective in deep or murky water, where most wavelengths of light have been filtered out by absorption. Because structural colors reflect specific wavelengths very efficiently and can be tuned to the wavelengths that penetrate deepest in the local water conditions, they can remain visible in environments where pigment-based colors would appear entirely washed out. Some deep-sea organisms use structural color in their signaling and camouflage systems for exactly this reason. Their colors are engineered, by evolution, for the specific optical conditions of the environment they inhabit. Human interest in structural color has grown enormously in recent decades, not just as a subject of scientific fascination but as a source of practical inspiration. Engineers and materials scientists working on structural color have developed surfaces and coatings that produce vivid color through nanostructure, colors that will not fade, that require no dyes or pigments, that can be tuned precisely by adjusting the geometry of the structures producing them. Applications ranging from anti-counterfeiting marks to architectural coatings to displays are being explored. The butterfly wing is becoming a blueprint. There is something quietly wonderful about that. The same principle that colors the wings of a tropical butterfly, the same physics of interference and diffraction operating at the nanoscale, is being studied by engineers hoping to build the next generation of color technology. Nature arrived at these solutions hundreds of millions of years before humans thought to look for them. It has been doing nanophotonics all along, inscribing it into feathers and shells and wing scales, hiding extraordinary physics inside ordinary beauty. The next time you watch a hummingbird catch the light, or notice the shifting shimmer of a dragonfly hovering over water, or look closely at the surface of an abalone shell, you are looking at engineering of the most refined kind. Structures smaller than the wavelength of light, arranged with a precision that produces color from nothing but geometry and the physics of waves. No pigment. No chemical. Just structure, and light, and the astonishing things that happen when the two meet at exactly the right scale. It is, in every sense of the word, beautiful. And the beauty, this time, comes with blueprints. There is a particular quality to the blue of a Morpho butterfly that stops people. Not just the color itself, though the color is extraordinary, a deep, metallic, electric blue that seems to pulse with its own light, shifting and brightening as the butterfly moves through the dappled light of a rainforest canopy. What stops people, when they learn the full story, is the reason for that quality. The reason it looks the way it does. Because the Morpho's blue is not blue in the way a bluebell is blue, or the sky is blue, or a piece of blue glass is blue. It is blue in an entirely different sense, produced by a completely different physical principle, one that has more in common with a hologram than with any pigment. The wing scales of the Morpho butterfly are built from a material that is, in itself, essentially colorless. Under an electron microscope, those scales reveal an architecture of extraordinary delicacy, rows of ridges, each one branching into smaller sub-ridges like the most intricate miniature fir trees, spaced at intervals measured in fractions of a micrometer. This geometry is tuned with nanoscale precision to the wavelengths of blue light. When white light strikes the wing, blue wavelengths reflect off the layered structures and interfere constructively, reinforcing each other, emerging stronger. Other wavelengths interfere destructively and cancel out. The result is a blue of unusual purity and intensity, generated entirely by the physics of wave interference. No blue molecule exists in that wing. The blue is, in the most literal sense, an architectural achievement. And it behaves differently from pigment blue because of this. As the wing tilts and the viewing angle changes, the interference conditions shift, and the color shifts with them, deepening, brightening, moving toward green or violet at extreme angles, always luminous, always somehow more alive-looking than a surface whose color comes from chemical absorption. The iridescence is not an accident or an embellishment. It is intrinsic to the mechanism. You cannot have the Morpho's blue without the angle dependence, because both arise from the same underlying physics. Hummingbirds achieve something related but distinct. The throat patches of many hummingbird species, those intense flashes of ruby or emerald or violet that appear and disappear as the bird moves, are produced by thin film interference in the microscopic structures of the feather barbules. The structures are arranged in slightly different ways from the Morpho wing, but the principle is the same: color produced by geometry, not chemistry. What makes hummingbird's structural color particularly striking is the way it switches so dramatically with angle. Face on in the right light, a ruby-throated hummingbird's gorget is an almost impossibly vivid red. Turn the bird slightly, or change the angle of the light, and the same feathers go dark, nearly black, the structural color condition no longer met. The bird appears to change completely from one moment to the next. It is the same feather. The same physics. Just a different angle of incidence. This quality, color that depends entirely on viewpoint, gives structurally colored animals a visual dynamism that pigment-colored animals simply cannot match. A red bird with red pigment looks red from every direction, in every light. A hummingbird with structural coloration becomes a different creature depending on how and where the light meets it. For a species where visual signaling plays an important role in courtship and territorial behavior, that dynamic quality adds information. The flash of color as the bird moves, the way it appears and disappears with changing angle, is itself a signal, and one that is much harder to fake than a static color patch. Chameleons extend this principle into something even more remarkable. the ability to actively and voluntarily change the color they produce. For a long time, it was assumed that chameleons changed color primarily by controlling the distribution of pigment in their skin, by moving pigment granules within specialized cells to concentrate or disperse color. And some color change in some species does work this way. But research published in the last decade has revealed that in many chameleons, particularly the most dramatically colorful species like the panther chameleon, the primary mechanism of rapid color change is structural rather than pigment-based. Beneath the skin's outer pigment layer, chameleons possess a layer of cells containing arrays of tiny photonic nanocrystals, microscopic crystalline structures arranged in a regular lattice. The spacing between these crystals determines which wavelengths of light they reflect most strongly. When the chameleon is relaxed, the crystals are packed relatively close together, reflecting shorter wavelengths, blues and greens. When the animal is excited or aroused, the cell containing the crystals expands, stretching the lattice and increasing the spacing between the crystals. Larger spacing reflects longer wavelengths. The color shifts toward yellow, orange, and red. The chameleon is not painting itself. It is tuning itself, adjusting the physical spacing of a nanocrystal array the way you might tune a musical instrument, and thereby shifting the wavelengths it reflects. The vivid color change from resting green to excited red or orange is the outward expression of a change measured in nanometers, happening in cells invisible to the naked eye. It is, by any reasonable definition, nanotechnology. And it has been operating in chameleon skin for tens of millions of years. The scientific and engineering communities have taken careful notice. Researchers working on adaptive materials, smart surfaces, and display technology have studied structural color mechanisms in biological systems with increasing intensity over the past 2 decades. The appeal is clear. Structural color offers durability that no pigment can match. Since the color depends on geometry rather than chemistry, it does not fade with light exposure. It offers precision. The color produced can be tuned with extraordinary accuracy by controlling the geometry of the structures. And it offers the possibility of dynamic, switchable color surfaces that can change their appearance in response to mechanical stress, temperature, electric fields, or other stimuli, by changing the geometry of their nanostructures. Artificial structural color surfaces are already in development for applications ranging from anti-counterfeiting features in currency and documents, where the angle-dependent color shift of a structural color element is essentially impossible to reproduce with standard printing, to architectural coatings that maintain vivid color indefinitely without the fading that affects conventional paints, to display technologies that produce color without the backlighting and filtering losses of conventional screens. The tiny structures of a butterfly wing are not just beautiful. They are, it turns out, pointing toward the future. Some of these structures behave, as the facts suggest, almost like miniature prisms or holograms, spreading light into colors through diffraction rather than refraction or interference. Diffraction gratings produce their colors by having light interact with very fine, regularly spaced features that cause different wavelengths to deflect by different amounts. The shifting rainbow shimmer of a CD surface, the spectral colors visible on certain feathers, the flash of color from a finely ruled metal surface, these all arise from diffraction. At the right scale, a physical surface can do with its own geometry what a glass prism does with refractive index: separate white light into its component wavelengths and send them in different directions. At still finer scales, structures can produce effects that begin to resemble the behavior of holograms, three-dimensional interference patterns recorded in a material that reproduce complex color and spatial information when illuminated. Some natural structural color systems create effects that depend on the three-dimensional arrangement of their nanostructures in ways that simple surface gratings cannot fully account for. The optics of the natural world, at the nanoscale, is richer and stranger than it first appears. And then, gently, the story of color moves outward from physics and biology into something broader. Into the human world. Into language, and culture, and the ways that different peoples across history and geography have carved up the flowing, continuous spectrum of visible light into named, bounded, socially shared categories. Because color does not arrive in the world pre-labeled. The spectrum is continuous, a smooth, unbroken gradient from one end to the other, with no natural seams, no places where one color definitively ends and another begins. The boundaries we experience, the point where red becomes orange, where blue becomes purple, where green becomes yellow, are not in the light. They are in us. And across human cultures and languages, they are drawn in strikingly different places. The simple fact that different languages carve the spectrum into different numbers of named colors has been documented across a vast range of the world's languages, and the variation is striking. English uses 11 basic color terms, the terms that are universal in common usage, each of which can be defined without reference to any other color. But many languages use far fewer basic terms without any deficit of descriptive power in the contexts those languages serve. Some languages have a single basic term covering what English speakers would call both green and blue, a category that linguists call GRU, and speakers of those languages navigate the world and communicate about color effectively without ever needing to mark the distinction that English treats as fundamental. The Piraha language of the Amazon, the Dani language of Papua New Guinea, and various other languages have been studied in detail, and they reveal that the number and placement of basic color categories varies enormously. Some languages mark a boundary between light and warm colors on one side and dark and cool colors on the other, as the most fundamental division in the spectrum. Others divide it at completely different points. The categories are not arbitrary. There are patterns and tendencies across languages that suggest some universal influences, likely rooted in the biology of human color vision, pulling color naming systems in certain consistent directions. But within those tendencies, the variation is substantial and real. What this means for the people who speak these languages is a question that has animated researchers for decades. Does the language you speak actually change the colors you see? Or does it only change how you talk about what you see, leaving the underlying perception identical? The answer, as the research has accumulated, appears to be somewhere in between, and more interesting than either simple option. The perceptual differences are real but specific. They show up most strongly in tasks that involve rapid discrimination, distinguishing between colors quickly, under time pressure, especially when the colors in question straddle a named boundary in the observer's language. They are less apparent in tasks that give more time, or that don't require the linguistic labeling system to be active. The effect appears to be strongest in the right visual field, which is processed by the left hemisphere, the hemisphere that houses language in most people, and weaker in the left visual field, processed by the right hemisphere. Language seems to influence color perception most when the language system itself is most engaged. This suggests that language and perception interact, that the categories language provides don't replace perceptual processing but run alongside it, sometimes shaping it, sometimes being shaped by it, sometimes amplifying distinctions that the perceptual system could make but would otherwise treat as minor. The color you see and the word you have for it are not as separate as they might seem. Each one reaches slightly into the territory of the other. And that reaching, that intertwining of the physical, the biological, the linguistic, and the cultural, is what makes color not just a phenomenon of light in the eye, but something woven through the whole of human experience. It is in the science of wavelengths and the poetry of names. It is in the wing of a butterfly and the grammar of a language. It is in the nanocrystals tuning themselves in the skin of a chameleon, and in the word a child learns for the color of the sky. All of it, somehow, part of the same extraordinary story. Color names are convenient fictions. Not false, exactly. They point at something real, something that exists in the physical world and in the biology of human vision. But they are not precise measurements. They do not carve the spectrum at its joints, because the spectrum has no joints. It is a continuum, and the names we give to regions of it are more like the names we give to regions of the ocean, the Atlantic, the Pacific, the Indian, useful and meaningful, widely agreed upon, but ultimately human decisions drawn across something that flows unbroken from one end to the other. The wavelength of light does not come labeled. 600 nanometers is not red. It falls within a range that most people with typical color vision would call red under ordinary conditions, in the absence of confusing context. But whether it looks distinctly red or slightly orange depends on the surrounding colors, the illumination, the individual observer, and the color vocabulary their language has given them for making that judgment. The boundaries of color names are soft, contextual, and variable in ways that the numbers on a wavelength scale are not. This is worth holding on to, because it helps explain why the relationship between color naming across cultures and languages is so complex and so interesting. The spectrum is the same for all humans with typical color vision, the same physical continuum, the same range of cone sensitivities, the same opponent channels processing the same kinds of signals. And yet the categories that different languages impose on that shared spectrum vary substantially, and the variation is not random. Researchers studying color naming across a large number of the world's languages have found that the spectrum tends to get divided in certain preferred ways, that some boundaries are more common than others, more stable across unrelated languages, more reliably drawn at similar points. The boundary between warm and cool colors is one of the most fundamental, appearing early in the development of color lexicons across many cultures. The distinction between light and dark, between colors that carry a sense of brightness and those that carry a sense of darkness, is another. These tendencies likely reflect something in the biology of color processing, in the opponent channels and the particular sensitivities of the visual system, pushing color naming towards certain natural divisions. But within those tendencies, cultures organize the color world very differently. The division between blue and green is one of the most variable boundaries in color naming across languages. English treats blue and green as fundamentally distinct, two separate basic color terms, no more similar to each other than red and yellow. Many other languages do not make this distinction at the basic level. In Japanese, the word ao has historically referred to both blue and green, and while modern Japanese now uses different terms for the two colors in many contexts, traces of the older usage persist. Traffic lights in Japan are technically green, but are sometimes described as ao, the blue-green word, a linguistic remnant of a time when the boundary was drawn differently. The most frequently cited cross-linguistic case of color term difference is the one involving Russian. English has one basic term for blue. Russian has two, голубой, which covers lighter, sky-tone blues, and синий, which covers deeper, darker blues. These are not shades of the same word, the way English has light blue and dark blue as modified versions of a single term. They are separate basic color words, as distinct in Russian as red and orange are in English. And as research has demonstrated, this linguistic difference has measurable perceptual consequences. Russian speakers are faster and more accurate at distinguishing blues that cross the голубой-синий boundary than blues that fall within the same category, while English speakers show no such advantage at that particular boundary. The category provides a perceptual edge. The word is doing work that goes beyond labeling. What makes all of this particularly interesting is that it runs in both directions. Language influences perception, yes, but perception and biology also influence language, shaping which distinctions languages are more likely to grammaticalize, which boundaries are more likely to be named. The two systems, the biological visual system and the cultural linguistic system, have been shaping each other across the entire history of human language and human vision, which is to say, across the entire history of what it is meant to be human. Color in human culture, though, extends far beyond the question of what we call things. It extends into what colors mean, into the emotional, symbolic, and artistic weight that cultures have attached to colors across thousands of years of history. Color has never been just a descriptor. It has always been a value. A signal. A statement about the world and about the person bearing the color. The use of colored pigments in human culture is ancient beyond easy imagining. Red ochre pigments have been found at archaeological sites in Africa dating back over 100,000 years, used in ways that suggest not just practical application but ritual and symbolic significance. The desire to make color, to possess it, to apply it to bodies and objects in spaces, appears to be one of the oldest and most persistent human impulses. For most of human history, producing vivid, stable color was genuinely difficult. The materials that could hold their color reliably, that would not fade in sunlight, bleed in water, or change with time, were rare, labor-intensive to produce, and often extraordinarily expensive. This gave color a direct economic and social meaning that it largely lacks today. The colors worn, the colors used in painting and decoration, the colors associated with particular social roles or classes, these were not aesthetic preferences in the casual modern sense. They were statements of wealth, power, and status, backed by real material cost. Ultramarine blue, made from ground lapis lazuli, a semi-precious stone mined almost exclusively in a single region of Afghanistan, was, for centuries, the most expensive pigment available to European painters. It was more expensive than gold by weight. Artists used it sparingly and strategically, reserving it for the most important figures in their compositions. The Virgin Mary's robes, in particular, became so conventionally painted in ultramarine that the blue itself became loaded with sacred significance, its visual weight inseparable from the cost of producing it. Purple, across many ancient cultures, was similarly loaded. Tyrian purple, extracted from the glands of sea snails, requiring enormous numbers of animals to produce even small quantities of dye, was so expensive and labor-intensive that it became associated exclusively with the highest levels of power. In Rome, it was the color of emperors. In Byzantium, the word porphyrogenitos, born to the purple, indicated an emperor's son born during his father's reign, a phrase in which the color stands directly for the power and legitimacy it had come to represent. These associations were not universal. Different cultures attached different meanings to the same colors, but within cultures they were often remarkably stable and consequential. The symbolic weight of color was maintained by social agreement, reinforced by law and custom, backed by economic reality, and sometimes backed by something more sinister. The history of pigment and dye is not only a history of beauty and status. It is also a history of toxicity, of the dangerous materials that were used for centuries to produce colors that could not easily be achieved any other way. Lead white, a brilliant, opaque white pigment used in painting and cosmetics across European and Asian cultures for over 2,000 years, is a lead compound, chronically toxic. Painters who worked with it daily were exposed to significant lead poisoning risk. Women who used cosmetic preparations containing it as a skin whitener over years were poisoned slowly and progressively. The beauty was real. So was the harm. Emerald green, the vivid synthetic pigment that became widely popular in the 19th century, contains arsenic. It was used in paints, in wallpaper, in artificial flowers, in fabric dyes, and the arsenic it contained made it genuinely dangerous in enclosed spaces, particularly in damp conditions where the pigment could interact with mold to produce toxic volatile compounds. Certain shades of fashionable Victorian green, in wallpapers and garments and decorations, may have been slowly poisoning the people who lived with them. Naples yellow, a warm golden pigment used in painting from antiquity onward, is a lead antimonate, another lead compound, with associated toxicity. Castle earth contains manganese. Cadmium pigments, still used today in artist-quality paints, are cadmium compounds, regulated carefully because of their toxicity in industrial contexts. Vermilion, the vivid red that appears in paintings and manuscripts across centuries of art history, is mercury sulfide. The history of color materials is threaded with beautiful poisons, colors that came with a cost that their users often did not fully understand or could not afford to avoid. The modern era of synthetic chemistry has largely solved the toxicity problem. Most contemporary pigments and dyes are far safer than their historical counterparts, while also making color abundant, cheap, and nearly universal. colors that were once the exclusive property of the wealthy are now available to anyone. The social meaning of color has shifted accordingly, becoming more personal and less fixed, more a matter of individual expression than inherited status. And in science, color has taken on new and critical roles where precision and accuracy in color representation carry consequences that go well beyond aesthetics. Scientific imaging depends on color in ways that are not always visible to the casual observer, but are profoundly important to the researchers who work with these images. When scientists visualize data, whether from medical scans, astronomical observations, microscopy, climate modeling, or any of dozens of other fields, they must choose a color scale to represent the range of values in their data. And those choices are not neutral. They shape what patterns are visible, what features are emphasized, and what conclusions seem to follow from the data. For decades, the most widely used color scale in scientific visualization was the rainbow color map, a scale running from blue through green, yellow, and red, covering the full visible spectrum. It looks vivid and comprehensive, and it has a certain intuitive appeal. But it has a serious perceptual problem: it is not perceptually uniform. The brightness and saturation of the colors in the rainbow scale vary in ways that do not correspond to the underlying data values. Yellow is perceptually much brighter than blue or red, creating an artificial visual emphasis on the middle of the data range regardless of whether anything scientifically important is happening there. Boundaries and gradients that exist only in the color scale appear as features in the data. Real features with certain value patterns can be hidden by the color scale's non-uniformity. Research has shown that scientists examining the same data visualized with a rainbow color scale versus a perceptually uniform scale, one designed so that equal steps in data value correspond to equal perceptual steps in color, can reach different conclusions, identify different features, and miss different things. The color scale is not a neutral medium for displaying the data. It is an active shaper of what the data appears to show. This is a striking example of how deeply color perception, with all its contextual sensitivity, its nonlinear brightness processing, its susceptibility to the effects of surrounding colors, reaches into domains where we might hope to be dealing with purely objective information. Even in science, even with the most rigorous commitment to accuracy, the human visual system brings its own properties to bear on what it sees. Design choices that ignore those properties introduce bias that careful methodology cannot fully correct. The recognition of this problem has led to significant work on perceptually uniform color maps for scientific visualization, scales carefully designed by combining knowledge of color science with knowledge of the specific data domain, ensuring that what the eye reads from the image corresponds as faithfully as possible to what the data actually contains. It is, in its way, applied color science, the knowledge accumulated from centuries of studying how the visual system works, brought to bear on the very practical problem of seeing clearly and accurately. Color, it turns out, is not just a quality of experience. It is a tool. One that can be used well or badly, with consequences that run from the aesthetic to the empirical. Getting it right matters. And understanding why it matters requires understanding something of the remarkable, layered, endlessly surprising story of what color actually is, a story that runs from the wavelength of light to the architecture of a butterfly wing, from the chemistry of a medieval pigment to the grammar of a language spoken on the other side of the world, from the firing of a cone cell to the moment a scientist looks at a map of the ocean and sees something that is or is not really there. All of it, color. All of it, real. All of it, somehow, part of the same continuous and extraordinary thing. Science is often portrayed as a process of replacement, old ideas giving way to new ones, mystery dissolving into explanation, the unknown shrinking steadily as knowledge advances. And in many ways, the story of color fits that picture. We have come a long way from the belief that white light was pure and simple, from the centuries when no one knew why the sky was blue or what a rainbow actually was. The mechanisms are understood now, in remarkable detail. The physics of wavelengths, the architecture of the eye, the opponent channels, the cortical pathways, the color constancy mechanisms, the nanostructures of butterfly wings, all of this has been mapped, measured, modeled, and described with precision. And yet color, at its deepest level, remains one of the most genuinely unsolved problems in science. Not because the mechanisms are unknown, but because the mechanisms, fully described, still don't answer the most fundamental question of all. Why does red feel like anything? Before arriving at that question, which is, in truth, one of the deepest questions in all of philosophy and neuroscience, there is one more thread worth following. Because even at the level of mechanism, even before we reach the philosophical abyss, there are things about color that still surprise researchers. Things that don't fit neatly into the standard account. Data visualization is one of them, in an unexpected way. We touched on this in the last section, the problem of color scales that mislead scientists by presenting data through a perceptual lens that distorts what the underlying numbers actually show. But the issue runs deeper than the choice of color map. It touches on a fundamental limitation built into the nature of human visual perception itself. Scientific instruments can detect and measure physical quantities across a vast range, far beyond what the human visual system can directly perceive. Radio telescopes detect wavelengths meters long. X-ray observatories detect wavelengths smaller than an atom. Infrared cameras detect heat. Magnetic resonance imaging detects the behavior of atomic nuclei in a magnetic field. None of these measurements produce anything that a human eye can directly see. To make them visible, to turn them into images that human scientists can examine and interpret, they must be translated into the narrow range of light wavelengths that human vision can detect, and mapped onto a color scale. This translation is unavoidable, but it is never neutral. Every choice about how to map unmeasured physical quantities onto visible color involves decisions that shape what patterns a human observer can detect. The visual system, with all its particular sensitivities and blind spots, its nonlinear brightness processing, its susceptibility to context effects, its strong response to certain color contrasts, and its insensitivity to others, all of this becomes part of the instrument. The detector is not just the telescope or the scanner. It is also the eye and the brain that examine the resulting image. Researchers in fields from astronomy to medicine to climate science have grappled with this, developing visualization techniques specifically designed to work with the properties of human color perception rather than against them. The goal is not to overcome the human visual system, that is impossible, but to align the structure of the visualization with the structure of perception, so that what the eye finds salient corresponds to what the data contains that is actually important. This is a field in itself, a meeting point of color science, visual neuroscience, and data science, and it is still actively developing. The limitations of human color perception are not just curiosities for vision researchers. They are practical constraints on the practice of science itself, shaping what can be seen and what can be missed in the data that the instruments of modern science produce. And then there are the edges of normal color experience, the places where the standard account of how color vision works begins to strain against reported phenomena that don't quite fit. The forbidden colors we discussed earlier, reddish-green, bluish-yellow, the hues that the opponent process system should make impossible, have been reported by subjects in carefully controlled experiments designed to push perception into unusual states. By stabilizing an image on the retina, preventing the small eye movements that normally prevent any single point from adapting completely, or by using other techniques that fatigue specific color channels selectively, experimenters have induced in some subjects the report of colors they describe as deeply strange, colors outside the normal gamut, colors they have no name for, colors that seem to contradict the rules of the system that normally produces color experience. Whether these reports describe genuine perceptual experiences of impossible colors, or whether they reflect some other artifact of the unusual experimental conditions, a confusion in the reporting process, a reaching for language where language doesn't exist, is genuinely unclear. The subjects are not fabricating. They are reporting something. But whether that something is truly a color experience outside the normal color space, or something else that resists easy description, remains an open question. What these experiments do reveal, beyond any doubt, is that the standard model of color perception, opponent channels, fixed primaries, a well-defined color space with known boundaries, is a good description of normal color vision under normal conditions. It is not necessarily a complete description of everything the visual system can be induced to produce under extreme or unusual conditions. The system has edges, and at those edges, things get strange. Now consider more carefully what the brain is actually doing when color experience occurs. The signals arriving from the retina are electrical, patterns of neural firing, varying in rate and timing across a large population of neurons. These signals are processed, transformed, compared, and relayed through multiple brain regions. At some point in this process, color experience arises. The neural activity produces, or gives rise to, or is accompanied by, the language here is inherently awkward, a subjective experience of redness, or blueness, or the particular warm amber of candlelight. Scientists understand, in considerable detail, what the neural activity looks like. They can measure which neurons fire, in which regions, at what rates, in response to different color stimuli. They can trace the pathways, identify the processing stages, describe the computations being performed. They can predict, from the pattern of neural activity, which color the observer is most likely reporting seeing. The correlation between neural activity and color experience has been mapped with impressive precision. What they cannot explain is why that neural activity is accompanied by any experience at all. This is not a small gap in knowledge waiting to be filled by the next experiment. It is a fundamental conceptual problem, one that philosophers call the hard problem of consciousness, and that neuroscientists increasingly acknowledge as something that sits outside the reach of current scientific frameworks. The easy problems of consciousness, explaining how the brain processes information, how it integrates signals, how it produces behavior, are hard in the practical sense of being difficult and complex, but they are tractable in principle. Given enough time and resources, they seem like the kind of problems that more research can solve. The hard problem is different. It asks why any of this processing is accompanied by subjective experience. Why isn't it all just information processing happening in the dark, without anyone home to experience it? Why does the firing of certain neurons feel like red, that specific, vivid, warm, insistent quality that red has, rather than simply causing the behavioral responses associated with seeing red, silently and experientially empty? No one knows. And more than that, there is currently no scientific framework that even points clearly toward an answer. The tools of neuroscience are extraordinarily powerful at describing the neural correlates of color experience, the brain states that accompany seeing red. They are not tools designed to explain why those brain states feel like anything. The question reaches into territory where neuroscience and physics and philosophy all run up against the same wall. This is worth sitting with, quietly, for a moment. Because it means that at the very heart of something as ordinary and immediate as looking at a red apple, there is a genuine, unsolved, possibly profound mystery. The wavelengths are understood. The cones are understood. The opponent channels and the cortical pathways and the color constancy mechanisms are all understood. And at the end of all that understanding, there is still something that is not explained, the felt quality of the experience itself. The redness of red, the blueness of blue. Philosophers call this the qualia problem, the problem of explaining the subjective, felt character of experience. And color is perhaps the clearest, most accessible example of it, because color experience is so vivid, so immediate, and so clearly tied to specific, well-understood neural mechanisms. You can trace the entire pathway from photon to neural firing pattern, describe every step, and still find yourself facing the same unanswered question at the end. Yes, but why does it feel like this? Memory and expectation add further layers to this already deep puzzle. We know that what you see when you look at a colored object is shaped by what you remember about that kind of object, by what you expect it to look like, by the entire history of your prior color experiences laid down in neural patterns across a lifetime. The color experience you have right now is not a raw reading from the present moment. It is the present moment filtered through everything that has come before. Which means that two people looking at the same red apple are not only potentially having different experiences because of differences in their cone cells or color processing, they are having different experiences because they have different histories, different memories of redness, different accumulated contexts within which the current perception is interpreted and completed. The red you see is your red, built from your particular biology and your particular life. And yet, remarkably, it communicates. You can tell someone the apple is red and they will understand you. The shared framework, the common biology, the common world, the common experience of light and objects and color that all humans share, is strong enough to bridge the individual differences and create genuine communication about color. Your red and my red are not identical, but they are similar enough, grounded in enough shared reality, that the word carries meaning across the gap. This is perhaps one of the most quietly profound things about color. It is one of the most subjective aspects of human experience, irreducibly personal, irreducibly the product of a particular body and a particular history. And it is also one of the most universally shared. It lives simultaneously in physics and in experience, in the objective world and in the private interior of consciousness, in the wavelength of light and in the felt quality of a moment of seeing. It connects us to the physical universe, to the sun that produces the light, to the electrons that interact with it, to the geometry of water droplets and butterfly wings and photonic crystals, and it connects us to each other, through a shared vocabulary built on a shared biology, navigating a shared world. The mystery at the heart of color is not really about color. It is about consciousness itself, about what it means for there to be an experience at all, for there to be something it is like to see. Color is just the most vivid, most immediate, most familiar place where that mystery lives. Every time you open your eyes and the world floods in with color, every red, every blue, every quiet gray dawn and every vivid summer afternoon, you are living inside a phenomenon that science has described in extraordinary detail and still cannot fully explain. You are having an experience that millions of years of evolution have refined and shaped, that your brain assembles continuously from nothing but electrical signals, that your language has given names to and your culture has given meaning to, that philosophers have puzzled over and physicists have measured and artists have devoted their lives to exploring. And it is happening right now, effortlessly, in the most ordinary moment of simply being awake and looking at the world. The colors are real. The experience is yours. The mystery is intact. And somehow, in all its strangeness and all its beauty, the world is still just quietly, luminously, there.